Hopefully, you're not having the kind of day we're having here. It's very wet and very rainy out in Kampala this last few days. So, welcome once again. With me today is Mulindwa Mohammed. Mulindwa is a quality assurance specialist here at the foundation, and he's going to be joining me for us to have a discussion around how do we reduce testing errors in the lab amidst the COVID-19 pandemic. And we want to touch on just the general lab space, looking at everything that could possibly happen in terms of reducing error. Uh, so just to be able to get started, Mulindwa, do you want to quickly say hi to everybody?
Good morning, good evening, good afternoon, everybody out there, depending on where you're coming from. Welcome to our webinar.
Okay, wonderful. So we've today, getting into today's conversation on this webinar, we're actually going to be looking at two standards. The first standard we're going to be looking at is the 22367, which actually just got revised in March of 2020. So it's pretty new, very, very revised standard. And basically what it is going to do is going to look at the elements of risk as it is associated with the ISO 15189, the 2012 standard. So we're going to bring both standards, ISO 15189 and ISO 22367, into our picture just to be able to have a discussion as to what the technical requirements are and how we can actually reduce risk. I always like to start off, because we're talking about standards and we're talking about the International Organization for Standardization and what they have done. Looking back into the Sustainable Development Goals. The Sustainable Development Goals, goals number 3 actually goes into looking at good health and our well-being. If you're going to look at that, of course, you want to be able to ensure the fact that we use the ISO 22367, and again, this was just revised in March of 2020 to a new version. And basically, the ISO 22367 looks into the reduction of error through risk management and continual improvement. So that's the element that we want to look at, reduction of error and ensuring that the patients continue to get the best health that they possibly can get. So when 22367 was revised in March, it's now called application of risk management to medical laboratories. Application of risk management to medical laboratories, and this bears heavily on ISO 15189, which is the standard that medical laboratories use to go on for accreditation. So just to be able to get started and to see how this all plays, the simple question I have is, when we look at an accuracy level of 99%, that usually an accuracy level of 99%, it sounds very high, sounds excellent. But actually what we're looking at here is a failure of 1%. When you look at the airline industry, just like if you looked at healthcare, for example, when you think about a failure of 1% on a jumbo jet that is carrying 387 people, that 1% suddenly becomes 100% to all the people on the jet. Yes, the accuracy level is 99%, sounds good, sounds phenomenal. But for that 1% where there is a problem, that's what we want to be able to spend time looking at. And it doesn't matter how small your laboratory is. You might have only 250 samples that you're working on in the whole year, in the whole month. But 250 samples, if you had 10% or 1% error, that is 100% to every patient to whom the error is associated with. So when we look at laboratory error, there's a few things that we're looking at that are of great concern. The first thing in laboratory error is we want to be able to ensure that we reduce the element of harm. Indirectly or directly to the patient, you want to reduce the element of harm. And so when we're talking about risk management in the area of laboratory medicine, we're basically looking at probability of harm occurring and the consequence of that harm. And we're going to look at some of the undesirable consequences of laboratory error when we go down the line. We're just thinking about the probability of harm occurring to the patient and what is the consequence of that harm occurring to that patient. In some cases, it has been as worse as somebody having the wrong leg amputated. Or it might be, in some cases, people have gone to surgery just to find out that they have the wrong patient in front of them. So reliability of laboratory results in medical settings is essential for correct diagnosis and positive clinical outcomes. So going forward, consequences of lab error are pretty grave when we think about what is going on. You have unnecessary treatment. It could be treatment complications because there was nothing in the first place. Failure to provide the proper treatment, delay in correct diagnosis, additional and unnecessary diagnostic testing because of lab error. Increased cost in time. You hold the patient unnecessarily, personnel efforts, and often poor patient outcome because of lab error. So let's look at percentages of lab errors. We deal with the different phases of testing in the laboratory. So I call this the pre-examination. In the pre-examination, this is what happens long before we even get into the pre-examination stage. You have issues of inappropriate test requests. The request form is a very important piece of paper. It's a very important document. It's a very important medium in terms of starting the whole process of testing. The request form, the order entry. Just identifying the patient and having the right sample for the patient is very key. The right sample collection, collecting the correct volume and the correct container. How is the sample going to be handled? What is the transport and storage going to look like? Then again, we have in the examination phase, and again, in that pre-examination phase, we're looking at errors well over 50%. And 50% of this is a very high risk of what could potentially happen to the client or the patient. In the pre-examination, sorting and routing of the samples, aliquoting the samples without proper mixture, pipetting and labeling could actually result in more error in the laboratory. And then we move on to the examination phase. 7 to 13% of the error occurs in this area, where it could be either equipment malfunction, it could be interference, or it could be just failure in quality controls. That would happen at this point. And then in the post-examination phase, testing is all happened, but we have erroneous validation of analytical data. Today, that is a very big thing, because today when we're talking about COVID testing, and we're talking about cutoff values, we're talking about the sigmoid curve, we're looking at trying to make decisions. When you have erroneous validation of the analytical data, yes, you have very grave effects here, because you could now result into a false negative patient. And in this COVID era, what you actually have is you're going to have an increase in potential contacts out there. Failure in reporting, addressing the report. Manual transcription. Manual transcription is also a very big problem in transcribing the results from one media to the next. If there is a transcription error, that will result into very big laboratory error. So these are just some things we're looking at. But then you have the total end phase of the laboratory error. These are errors in healthcare when we lead to actual or potential adverse outcome for the patients. And there is data that is already published. And this is really important to look at the percentages that we're looking at here. We have 24 to 30% of laboratory error have an effect on patient care, while actual harm or potential harm occurs in 3 to 12% of that population group. That's a very big population group when you're thinking about it. And what is important to note when we think about laboratory error is the fact that for each patient to whom there is an error, that is 100% of error within that laboratory. That is 100% error to that particular patient. So, going back onto ISO 15189, there are actually 3 big areas in ISO 15189 that have to be looked at. The first one being the pre-examination processes. And when we go out of the standard, it basically says the laboratory shall have documented procedures and information for pre-examination activities to ensure validity of results of examination. Validity, that's a very big thing. Then the next big phase is the examination processes themselves. This is the area where we actually look into the verification and validation, the identity of the persons that are going to be performing the test, and the processes that shall be used to record the testing processes. And then we have 5.6 of ISO 15189, which actually looks into ensuring the quality of examination results. In looking at quality of examination results, this is very big because it actually determines whether the actual result is going to be useful or whether there is error in that result. The laboratory shall ensure the quality of examination results by performing them under defined conditions. The conditions must be known. The technical personnel actually executing the test must actually be competent in doing the test. And then finally, of course, we have the examination process, the post-examination process. In this process, we are basically asking, now that the examination has happened, are all the procedures used? Are they the best procedures? Do we have authorized personnel? Do we have people who have the competencies to be able to review these examination results before they actually go out? So these are areas of potential error in laboratory that we want to visit. And just as I go on to call in one of our associates, Mulindwa Mohammed, to have a discussion in the next series, we've actually broken this into 2 or 3 sections. We want to look at external quality assurance, IQC, lot testing, validation, verification, and personnel as one area that we want to have a discussion. And then we look at measurement uncertainty, calibrations, the trend analysis that are involved, review of results, again, back to the standards, and then interlaboratory comparison. Then there is a big, nice piece that just sits under there, is actually the post-market surveillance area. So once all of these things have happened, product is in the market, who actually goes back to ensure that what we have in the market is actually what we're going to bring to market. So I'm going to welcome Mulindwa at this point to take us through the first phase. Mulindwa, welcome. And it's actually great for us to be sharing the platform today. I think I'm actually excited about this. So please, let's go in. Let's talk about external quality assurance, interlaboratory comparisons, and all of those beautiful things that could reduce lab error. Welcome.
Thank you so much, Professor. It's a pleasure to be here to talk about some of these things. I'm called Mulindwa Mohammed once again, and like Prof said, I'm a quality assurance specialist here at the foundation. Just picking up from where Prof left off, Prof has talked about what are some of the errors that we encounter, the processes where we encounter these errors. So for me, it's going to be very simple. I'm just going to talk about where are these errors coming from, what are the sources, and how can we reduce the errors. So there are basically 3 types of errors when you get to look at the big picture of errors in clinical laboratory medicine. These types are human errors that arise due to human interaction with the samples and the assays. There's what we call the assay error itself, because you as the human being, everything might go right, but there might be some challenges with the assay that you're using. And also there's what we call environmental errors. These are the errors that come due to where you're doing the testing from. So just to dig a little bit deeper into each of these errors, we know that for human errors, it can be anything. It can be lack of training. It can be lack of mentorship. It can be incompetence. It can be non-compliance to set procedures and requirements. It can also be due to negligence, because we cannot underplay the effect of what we call negligence and lack of proper due diligence when you are doing your work. It can also be an ethical issue, because we've seen a lot of things happening in the labs, but it's not because the people are not competent, but it's because there is some sort of degree of unethicalness. And then there are very many other sources where errors can come from. So when you get to look at what we call the assay error, the assays themselves can come about with some imprecisions associated with them. It might be an inaccuracy issue, whereby the assay has some degree of inaccuracy that doesn't meet the requirements. It can be poor maintenance by the labs. It can be lack of servicing of the equipment. It can be the quality of what you're putting into the equipment. So all these things can bring about what we call assay errors. And then there is what we call environmental errors. So environmental errors these are due to the conditions of the environment themselves, because if the assay is not done in an environment where its manufacturer says it should be done, you expect to get some errors at the end. It can be a space inadequacy. For example, some equipments, they need a minimum of some space. And if you don't give them that space, there will be issues. There can be cross-contamination. For example, you're conducting some activities that are incompatible, and then you encounter some cross-contamination. And I think one very critical example that I can give here is the storage, where we have been storing our reagents. Some laboratories store reagents with the samples, so you kind of sort of get contamination, and also in microbiology. So some of those things have to be put into consideration. There's what we call biorisk issues. These are issues that are related to biosafety and biosecurity of the equipment and the assays that you're conducting and the work that you're doing. There are some sort of errors that can come if you don't actually take care of that component. And then other errors that like we shall see going forward. So how can we reduce these errors? Like Prof said, it can be 1%, but that 1% means a lot. But if you reduce it through doing simple things like participating in external quality assurance, that error has a chance of not happening. Why? Because in external quality assurance, we are looking at where are you participating. So in terms of participation, there are different platforms where you can participate. There are some platforms where they do sequential participation. A central provider sends one sample throughout different laboratories, and you all come up with some sort of results, and then the evaluation is done. There is simultaneous participations. For example, in those EQAs where different samples are sent to different labs, but then a centralized evaluation done. There is interpretive, where they just give you a sample and they tell you to interpret the results. And then there is sample review, whereby the lab does the testing, and then it sends it to a central lab to verify what it did. And then there is what we call split sample. This was a mistake by simple sample, but it's split sample. You get one sample, then you distribute it between the labs. That's called split sample EQA. So when you participate in any of those, you're participating in what we call external quality assurance. But one most important thing to note is that the reports that come from that participation, they have to be analyzed and reviewed. So you have to review whatever you've got as feedback, and then come up with root cause analysis and corrective action in case there is any failure that you've encountered. So that is really important. That is one way that the lab can use to minimize error. Another one is internal quality control. This is a very common phenomenon whereby the labs run samples periodically. Some tests require that you run internal quality control samples on a daily, on a weekly, on a monthly, so depending on what the lab has documented. So it's important that you run internal quality control samples just to verify that your test is performing within the defined limits. Because if there is any failure, you can come up and do what we call troubleshooting. So it's important that you define the frequency of how often you're going to do the internal quality controls. And then whenever you do the internal quality control, you come back and review. Have we met our criteria? Is it still within the limits that we set? If it is out of the limits, you have to look at the trends and say, how often has this occurred? Or this one time that it has occurred, is it warranting corrective action? So you come again, you come and do what we call an investigation or root cause analysis, and then perform corrective action. So it's important that you do internal quality control because it helps you ensure that your results are reliable. And then there is this beast called load testing. Sometimes different laboratories in different countries do this differently. Some labs say they want to do load-to-load testing, whereby they just get an old batch, run internal quality controls on the old batch, they get the new batch, run internal quality controls on the batch, and then they do what we call comparability comparison. So in case the lab does that, it has met the requirements, in case it's what it defined. Some other labs do it differently. Some other labs, they use other statistical process control analysis to run the sample, that the reagents that have just come in, and then do statistical analysis on them. to just ask themselves, is it still within the limits? So that is also acceptable. However, some of the most important things to note is that you have to look at the manufacturer's insert. The manufacturer's insert tells you exactly what you need to look at, what the manufacturer set as the limits, so that when you're doing what we call load testing, you are doing it within the manufacturer's set criteria. And also you have to have a procedure for conducting load testing, because this procedure is going to guide you on a lot of things on the how we are going to do it, but also what is the acceptance criteria that you're going to use. That is very important. And then you have to ensure that you maintain the records, because it is these records that you have to go back and look at when you're doing trend analysis. If there is a lot that came in, and let's say it was within a 2 SD standard deviation, and now we have got a lot that is 1 SD, we can make a decision and say, let us use this current reagent, irrespective of whether the other one passed or not. So it's important to maintain records. One more thing that the lab can look at is method validation, stroke verification. It's important to know when to validate your tests, because a lot of times the manufacturers give us instructions that this is how you should use my assay. But when it comes to the lab, due to different reasons, some of the reasons might be resources, some of those reasons might be the level of the staff that are in the lab. Some labs have really high level staff that can modify tests. So in case you modify, you have to validate. In case you change anything that the manufacturer said you should do, you have to validate. In case you don't change anything and you are bringing this assay into the lab, you have to make sure that you verify its performance before you can accept it into your normal routine. So you do that to just have some degree of confidence that says that the established error limits for this test is ABCD and it's acceptable. And that is one of the ways you can reduce errors. So you have to stick to manufacturer claims. What did the manufacturer say that the assay does, and what are the performance characteristics that he used, so that you can also establish based on your performance characteristics. And then you evaluate whatever you've come up with against the criteria. So if the manufacturer said that my performance criteria was, let's say, my performance characteristic was sensitivity, and I passed the assay by saying that it's 90% sensitive. In your lab, if your acceptance criteria is 90%, that's what you verify. But if your acceptance criteria is 100%, you have to make sure that whatever results you've got, you evaluate them based on the 100% defined by the lab. And that is where manufacturer claims come into play. But another important thing is not everybody approves the verification report. You have to make sure that whoever approves the verification record is competent and actually has the best interest of the patient at heart. Because you might want to accept something just because it has been either pushed onto the lab, you have no other options, but that is one thing that the lab has to look at: who approves verification reports. They have the right competencies and the right criteria. So it's important to also look at personnel competence, because usually they say garbage in, garbage out. It is the staff that is going to actually use whatever has brought in to release the result. So if the competence of the staff is not taken care of, the results that are going to come out are going to actually be unreliable. So you have to make sure that the staffs are trained, and you have to define the frequency of how often you do competence assessments. Some labs do it differently. Some say that when you come in, initial competence can be done within 6 months and then 1 year after. But some labs need more frequent competencies than that. So that's up to the lab to define. And then other things like the scope, where is the competence going to start from, and the acceptance criteria, and also the competence records. You need, whenever you run a competence assessment, you need to be traceable to a certain result that has been released. Thank you.
Mulindwa, you touched on some really critical and some very important things here. And just before we look at the next set of elements that can help in reducing error, let me just take a second here, because when we're talking about EQA, we're actually talking in its own definition, external quality assurance. we are actually allowing somebody from outside to compare us with another group of people, but also to compare us against our own platform in a different range. And then there is, of course, the element of internal quality controls, which is critical because it allows for us to be able to build the element of trend. It allows for us to be able to look into the element of bias, which is crucial in terms of how we see that there is a potential error coming down the line. But just as you close, you touched on two very important words in ISO 15189. And it's also very important when we're talking about the ISO 22367, which looks at risk. That is the element of verification, ensuring that what the manufacturer said you're going to get is what you actually get in your systems. And the element of validation, which basically says if you're going to be acting out of the standard approved requirements, then you have to conduct validation. Those two words in medical laboratory practice are used very interchangeably so often. However, they mean very different things. And if they are not looked at carefully, that's a source of big error. Whether you're doing verification, ensuring that the manufacturers, whatever the manufacturer told you is what you get, or whether you're doing a validation because you are acting out of a standard method. But as we go on, we're also going to have the discussion about measurement uncertainty, which is also another big element that has a huge play when you're talking about verification and validation, but also when you're looking at your relationship, when you're talking about external quality assurance and internal quality controls. That element of measurement uncertainty is an area which we need to look into because it has huge relevance. And just if you want to piggyback again some more, at the beginning, we spoke about the sigmoid curves and all of those kinds of things. When you're talking about cutoffs, the big conversation today when we're talking about COVID-19 and the PCR tests is trying to figure out, in terms of verification, the manufacturer has a cutoff that they have for reading their results. In terms of validation, if we want to change where we want to read those results from. But again, in terms of understanding what calibration is involved, what calibrations are necessary for the pipettes and the other things that we're going to use. So just before, as we go into the next area where we want to talk about measurement of uncertainty and calibrations and these kinds of things, if you don't mind, Mohammed, please jump back in and take us through the next sets of errors.
Thank you so much, Prof. So, like we said, Prof talked about measurement uncertainty and what exactly this is. You are just establishing the degree of doubt that you have about the essay result that you're releasing. So if you release a result, let's say it's a CBC done by a certain equipment X, you want to establish and say, whenever I release a result from this equipment, this is the degree of doubt, this is the range of results where the actual results will fall. So it's a very important requirement that actually laboratories establish their measurement uncertainty. Because with the measurement uncertainty, you are establishing and you're giving the clinician confidence that the patient might be symptomatic or asymptomatic, but there is this limits that the result might actually fall within the critical or non-critical. So please look at it accordingly. So it's important for the lab to know what exactly they are measuring, because if you don't know what you're measuring, you cannot establish the measurement uncertainty or the degree of doubt with which that equipment is producing. And then also you have to talk about the quantitative steps, because measurement uncertainty in itself is only associated with a quantitative step or a quantitative method. So if a method is a quantitative method, you have to establish its measurement uncertainty. Or if it is an essay that is qualitative, it gives a result that says you're either positive or negative. But to get that result, there is a measurement step. You have to get the measurement uncertainty of that measurement step. And then also it's important to establish the sources of uncertainty, because if you don't know them, you cannot quantify the uncertainty. So it's important for the lab to define the sources where this uncertainty is going to come from and establish a procedure to be used to calculate the uncertainty of measurement. Now, uncertainty of measurement is particularly important whenever you're using any tool in the lab. For example, a pipette, a measuring tool like a thermometer, a measuring tool like a weighing balance, a measurement tool, anything that you can use to measure, you have to establish its measurement uncertainty by ensuring that its uncertainty is available. So that is one of the areas. And then we look at trend analysis. Trend analysis is one way that the lab can use to identify any variations in its processes. So you have to ensure that the data is collected and maintained properly. You have to look at the trend analysis frequency. How often are we going to look at these trends? Because if you set shorter time frames, you might not get the actual trends that you're trying to look at. So if you set, can I say, an amicable timeline, that can give you a better picture of how your process is performing. So it's important. And also you have to establish acceptance rules to say that the process varies between a certain limit, upper limit, and a certain lower limit. And if it exceeds any of those two limits, you come and do root cause analysis and corrective action. We cannot talk about error without talking about review of results, because whatever you're giving out is what is going to be used to manage this patient. So before you give it out, there has to be somebody to give it a second eye. And this somebody that gives it a second eye has to be competent. There has to be a criteria that this somebody is going to go through to say that when I'm looking at a result, I'm looking at this number of things. And some of these things can be completeness. Are the results complete? If you say that the patient has this number of grams per deciliter of hemoglobin, did you actually put it in grams per deciliter or in millimoles per liter? Because different clinicians, they actually interpret those results differently. So you have to make sure that the result is complete in its SI units, and what the clinicians understand is what you're giving them. You have to look at the transcription errors. If the machine says that it is 9, is it still 9 on the paper that we're giving the clinician? You have to look at are the persons identified that conducted the actual testing. In case of anything, can we trace back to where the issues are coming from? You have to look at a lot of things, but in totality, there has to be a criteria and issues that the reviewer is looking at. And also, like we said, there has to be authorities. If you're going to do any amendment in any result, we have to know who is authorized to amend a result, who is authorized to access that result that you are releasing, and who is authorized to make any necessary changes onto that result. Interlaboratory comparison, the reason why this one actually came after the review of results. Some people may think about interlaboratory comparison in the same bracket like EQA, and that is in a certain sense true. However, interlaboratory comparison can be thought about as the alternative to the other processes that we talked about. If a lab does not have the resources to participate in EQA, there is an alternative called interlaboratory comparison, whereby it can just participate with its peers. And there has to be a kind of sort of a coordinating lab to just coordinate the exchange of samples, and all coordinating body. It can be a lab, it can be an organization, just to coordinate the exchange of samples and review of results, and also follow the ISO standard for statistics to make sure the interlaboratory comparison meets the requirements of the ISO 15189. So in case you participate, you need to make sure that the logistics of the program are well defined. There has to be statistical analysis, like we talked about, and of course a criteria that says if you get these results, you passed. If you get these results, you failed. So go ahead and do root cause analysis and corrective action. Thank you so much.
Indeed, Mulindwa, you actually, again, this is actually, I think this is one of the more critical webinars that we've had when we're thinking about elements around both standards, ISO 15189 and ISO 22367. But just two things I want to follow up on, because you really touched on some really important things. The element of interlaboratory comparison. The element of interlaboratory comparison is one that we see very frequently on the African continent. And of course, for obvious reasons, the element of cost and participation in an external quality assurance program. However, if we're going to get into an ILC, interlaboratory comparison, it is very important that there is a set of criteria that is set before we even start exchanging samples or testing within our labs. Because when you get a result, let's say you had a quantitative result, the fact that you had a hemoglobin of 12.3 does not suddenly mean that it's acceptable if there wasn't a predefined criteria that was actually agreed to before you did the interlaboratory comparison. So when we talk about interlaboratory comparison, it is key that our laboratories actually understand the requirements before you get engaged in interlaboratory comparison. It's not just about looking at what is the value of the result when you have it. But also, let's go back to measurement uncertainty, which is another area where there is a huge debate, and the standard ISO 5189 is very clear in that particular area, and that is the element of the difference in qualitative and quantitative results. And I think you did a phenomenal job touching on that, because people think that there is no measurement uncertainty in a quantitative result, and which is certainly not true. As long as there is a measurement step in that quantitative result, then you have to have a measurement uncertainty that is going to be calculated. It is very direct and obvious that there is measurement uncertainty when you're talking about quantitative results. So we need to have a good understanding of the relationship between the quantitative result and the measurement uncertainty of the final result, and a quantitative result with the measurement uncertainty at any point where there is a measurement and there is a measurand that is going to come out of that measurement. I think those are two very important things to include. How do we get involved in interlaboratory comparison? But now, just before we jump onto the last piece, which is the post-market surveillance, that's another area. In most of our cases, what we have is we get product, we get reagents that come into our systems. And we do, as you rightfully said at the very beginning, we do a lot comparison, a lot verification, and then we go to work. And then we don't get a chance to be able to ensure that from one lot number or from one batch, we have the same thing as the manufacturer actually told us would have. So please, why don't you jump into what happens with the post-market surveillance?
Thank you so much, Professor. So post-market surveillance is one of the things that actually is not done in most of the countries that are conducting laboratory testing. But it is something that is really, really very important, because for you to know where you're going, you need to know where you're coming from. So it's important that the country or the lab works with the different authorities to actually go ahead and do what we call a post-market surveillance. So we've procured all these reagents to conduct our complete blood count test. But the thing is, how was complete blood count test performed over a certain period of time? And what are the decisions that we need to make sure that we improve the services of our clients going forward? So it's important that we have a plan. A plan is very important in post-market surveillance, for you to know what exactly are we looking at, what are the activities involved, who is responsible for what, and then what are the outcomes of each of the activities. It's really important, because now if we have a well-defined plan, we can evaluate whether the objectives are going to be met. And also, we need to be mindful about how we design the post-market surveillance and how we're going to do the sampling. Because if you're going to do a post-market surveillance, for example, for laboratory reagents in general, we need to design a mechanism that takes care of the different factors that affect testing and supplies. Some of these factors can include geographical locations. We need to consider how are the different geographical locations performing on this essay. We can consider what are the different levels of the laboratories. For example, I can just give an example where in Uganda we have the high-level labs, we have the middle-level labs, and then the low-level labs. How is it performing? So there are different aspects that we can look at when we're designing the post-market surveillance, and that has to come in really handy. And also, we cannot do a post-market surveillance without actually doing a risk-based planning, because it is these risks that are going to affect the outcome of the report that we are going to give. So we have to do a complete risk assessment that takes care of the different processes that we're going to look at in the laboratory testing process, and also in our post-market surveillance process, to harmonize all the risks, and we say these are the mitigations that are going to take care of all the risks. And also, it's important to be aware that data collection is not just gathering data, but the how is very important. How we're going to gather data that actually makes meaning. It's very important to gather the right information, not just to gather any information. So how you gather the data and how you analyze the data is actually going to be very key when you're preparing the report at the end. And also, when you come up with the report, we need to be aware of who are we going to present this report to. Because we've seen in some areas where some of these nice things are done, but then the recommendations are not taken to the right audiences. So if we have recommendations, who are the people that are going to actually listen and work on some of our recommendations that are coming out of the post-market surveillance? So all these things are really important.
This is really key, and it's very interesting that we're in the COVID era now, and we don't have time to talk about post-market surveillance in COVID era, because what we're actually looking at today is we have what is called the Emergency Use Authorization, which most people have come to know as the EUA. And what happens at post-market surveillance is really what is happening now with the Emergency Use Authorization, because basically we are actually trying to ensure that the products and commodities that come to the market using the EUA literally have not gotten enough chance to be tested. So while in the post-market surveillance, we're ensuring that what comes to market is what originally came and we're getting the same quality, with the Emergency Use Authorization, we're ensuring that what actually gets into the market gets a chance to be able to collect the right amount of data so that there is certainty in the reliability of the results that we get. So we have these two extreme spectrums. We have Emergency Use Authorization before we get into the market, and then we have the post-market surveillance after we've been in market and what is going on. So those are some areas that we need to look at. And these are some very simple things that we spend some time looking at to get this conversation on how to reduce laboratory error. But one of the things, because we're in the standard space, that we want to let everybody know is with every new standard that is coming out, the element of risk and risk assessment is going to be a big part of it. And basically, when we talk about risk and risk assessment, what the standard is actually looking for, the standard is basically saying, what are your opportunities for improvement? So it's basically the opportunities for improvement, the OFI, as most people are going to come to know it in the industry. OFI is very crucial in ensuring that you run a management system that allows you to continue to give reliable and quality results going forward. So in terms of talking about laboratory error and in terms of looking at the things that have happened and the things that could happen, the following 10 points that we looked at today are some of the key areas that all our laboratorians, everybody who is scientists, and this is not only about medical diagnostics. If you're in the food industry, you need to be thinking about some of these errors. You need to think how they work. In any of the calibration industries, if you're doing any measurements, these errors are very significant. If you're in the pharmaceutical industry, this is even big. And when we talk about 22367, it actually has a huge bearing on the ISO 14901, which actually looks at what happens in terms of device. And the change in 22367 actually has a huge bearing in terms of our pharmaceuticals and our devices. So this is a space that we need to look out for, because again, as I said at the very beginning, you could say the accuracy is 99%. And it sounds phenomenal when you say accuracy is 99%. But that 1% is a specific patient to whom that 1% error is actually 100% to themselves and their family, and it's a huge cost. It's a huge burden to them. So laboratory error is something that we want to be able to reduce. We want to reduce it tremendously. And I'm going to go back to what I initially said when we were launching this idea. When the IOM, the Institute of Medicine, did its report in 1999, if I remember the date well, to err is human, one of the big things that we're looking at that point was how can we ensure the safety of our patients? So at this point, just before I take you in and talk about what we need to do to stay safe in the COVID era, if there are any comments, questions, anybody with some remarks they want to make at this time, we'll be very glad to have you. Anybody with some comments or remarks? Let's see. This was one to Mulindwa. It says, thanks Mulindwa for a great overview. My question is there any electronic medical record that can track all the errors and improvement under your environment? A lot of work needs to be recorded, like reports, forms for tracking, the elements, load testing. In essence, he's regurgitating a lot of the things that we've just mentioned. But the element here is there any electronic medium that we could use to be able to track on this?
Thank you so much, Sharif. I think this is a very important question. And I think what you've just given us is an idea of the next big thing that maybe we might look at for the labs. Currently, there is nothing that we know of, but I know that at a Global Healthcare Public Foundation, this is something that we can help the labs come up with, and we can help the labs actually implement this very well. So thank you so much. This is a very nice idea, and I think we're going to look at it.
Yes. I think one of the things we've been looking at the foundation is coming up with simple scientific modules that we can help the lab be able to track what is going on. And the element of tracking error is very key in improving the quality of the laboratories. Any other comments, questions, concerns? There is a hand up. If you would just unmute and go on and speak. Sharif, that was you again. Is there anybody who has a comment or has a question? Well, I think this was a very simple, straightforward, direct presentation. Sharif, are you speaking?
Thanks everyone. Thanks Mirinda for the wonderful overview, and thanks for the work, Mr. Professor. I think you have tried to give a very nice overview, and I was so impressed by the medical lab. So my idea, I think I actually tried to comment. It was about any system which can record all this stuff. And that was my query, and you have answered it. Yeah, thank you.
Okay. Thank you very much indeed. Thank you, Sharif, for the comment and for the question that you placed. As we already mentioned, those are areas that we are looking at, because it certainly strengthens the lab, and it allows them to be able to see potential for error in real time. And just before the results go out, if something can be done about that error, that is always very important. Any other questions, comments? We are just inching into the top of the hour for this presentation. We try to keep them under 40 minutes and take a few questions and comments for another 15 or so minutes. So if there are none, again, it's always a pleasure. What we have here today, of course, I never want to end any of this without telling you to continue to please wash your hands with soap and running water. In the absence of some soap and washing and running water, please sanitize as many times as you possibly can. It's important to keep sanitizing. COVID-19 is a respiratory disease. It is a droplet disease. It is all over the place. In one of my earlier presentations, I said 10% of the infection rate is going to be environmental. It's floating out there. So if you wear a mask, there is a chance that you're going to reduce the opportunity to be infected. So please do wear a mask, and social distancing, in spite of the fact that you have a mask, is very important. We're doing it everywhere we are, even on the planes as we travel. Thank you so very much indeed for listening in to us. You certainly could reach us, phone, Twitter, any of the handles that you can see there right now. You could certainly reach us at that. I wish you a wonderful week. And thanks, Mulindwa, for joining me today. I think I really like the way this went. I think we're going to do some more of this.
Thank you so much.
Thank you. Wonderful. I look forward to seeing you next week. Same time, same place. Have a wonderful rest of the week.